Thanks for joining me today for this short talk on update some ongoing research where we're bridging multiple lines of evidence to quantify plant phenology in Southwest area ecosystems. So PhenoMed is the ongoing study that we've had, or ongoing study since 2010, that was really designed to bridge plant phenology and meteorological observations as well as soil moisture. One of the driving forces of this project is to identify those environmental factors which are most important to trigger plant responses, seasonal transitions. So the initiation of growth, canopy development, the production of fruits, the production of flowers, as well as the end of the season, because that is not well known or understood in water limited systems in the Southwest. So this mechanistic understanding is what we're after, but we're also interested in how then that understanding can be applied and modeled at different spatial and temporal scales. So that involves a multi-scale assessment of these phenomena at different levels using different data sets that have different temporal resolution, so the frequency with which they're collected, the spatial resolution, which is essentially differences in size of the imprint of the observation, as well as the spatial extent. So there's grain extent and then the temporal frequency. And each of these data sources have a different currency. So what I'd like to share with you today are primarily links between various layers in this multi-scale assessment, provide an update on some recent results, as well as to share with you where we're going next with future research. So the links that I'm going to be focusing on today are those between field measurements and phenocams, which are digital cameras that are mounted in the field and are taking multiple images a day with a digital camera, as well as the then link between the phenocams and high spatial resolution data that's collected with an unmanned aerial system or an unmanned aerial vehicle, UAVs. So just to provide a depiction of what the field data look like as part of this study, this is one site. It's a May 2012 high-resolution image. What is red in the image is actually green, so it's photosynthetically active. Those are the shrubs, and the green in the image is the dominant grass at this particular site, which is black grama, Bouteloua areophyta. So the yellow triangles represent the plant observations. We have between 30 and 34 individual plants at each of 5 sites at the Jornada, where we're monitoring phenology status as well as percent greenness every week. The yellow stars at the top and then here in the central portion of the image are the 2 cameras that we have mounted, and you'll see an image of those in a few moments. So primarily today I'm going to be talking about the patterns in black grama, the dominant grass at this site, and honey mesquite, the dominant C3 deciduous shrub. So these are what the cameras look like. We started out with a simpler setup here, which is a $100 plant cam, that we then graduated to a more sophisticated system that has higher resolution, as well as the ability to transmit the images wirelessly. So we're getting repeat images every day from each of these cameras. And what we've done to derive data from these cameras is identified areas in each image that correspond to the species of interest. So in this case, there's the mesquite here, and then the mesquite's in the foreground on this image, and then the white represents black grama. So what I'm going to be showing you today are species-specific greenness patterns that we are deriving on a daily basis from these repeat red, green, blue digital images. We're also comparing the greenness patterns from the cameras with what we're observing in the field. So we're using standardized protocols that were developed by the National Phenology Network, and for the comparison with the phenocam, we're focusing on Canopy Greenness. So it's a categorical estimate that's evaluated for each plant and each week. So when we correlate these two values, the field estimate and what we're getting from the camera on that day for that species, we're getting positive and significant correlations between the two measures. So what this provides us is kind of confidence in the data that we're seeing at a final temporal resolution of every day with the cameras. They're reasonably representing what we're seeing in the field. So this is the green and the brown are the lines from the respective species, green being the grass, brown being the shrub, and then the bottom graph represents daily rainfall. And so these are the types of data that we can derive from the phenocams, and this is just a snippet of the subset of the data from one camera. And then once we have those lines, those greenness lines, and they're the results of a regression on the multiple daily values that we have, we then can derive seasonal metrics. And in this case, we're using a simple thresholding technique. And so from this, we are able to identify the start of the season from the camera, the end of the season from the camera, and then the length of the season, the date of peak, and then the peak magnitude. So when we then and another way of looking at this is the start of the season. So in the last slide, we talked about season length. And in this particular case, we're looking at the start of the season. So in each case, the field observed values are on the x-axis, the camera-derived values are on the y-axis, and the numbers are the day of the year, starting in January 1 as being the value of 1. And so the values for the mesquite shrub are in gray. And so at both the beginning and the end of the season, we're getting very good correspondence between what we're seeing in the field and what we're seeing from the cameras as representing the start of the season. But for the grass, the black grama grass that we see in the black, the start of the season is showing up much later from the camera than what actually is happening on the ground. So the ramifications here are that with the season, with the phenocams, we are grossly underestimating the length of the growing season for the black grama grass. And that might not be the biggest ramification, the biggest source of might not be the most problematic. The most problematic case would be potentially if you're using those start seasons derived from cameras to evaluate the role of the environmental parameters in driving that change. So you're missing the actual start and you're modeling something else. So we are leveraging high resolution imagery collecting, this is a 50 by 50 meter area, the same spot on the ground in 3 years. The idea with using these data is to then facilitate the link between what's observed on the ground and then what can be seen via the satellite. So we have species-specific classifications for these images through time. And I'm just going to highlight an example here rather than go through the whole time series. In 2013, this is what it looked like. So it's dominated by grass. The brown are the mesquite shrubs. And then we're tying in then from the phenocam the greenness profiles that we know from the field data are representing what we're seeing in the field. So this particular date that this image was collected this is where we were in terms of January to January. So this was collected in September. And if we fast forward one more year, the same sort of phenomenon. So the idea is that we know what the plants are doing from the cameras. We know where they are and how much of them occurs at a given location with the UAV imagery. Where are we going and why are we doing this? We're basically using all of these data to enhance our interpretation of time series data that can be derived from Landsat, which are the pixels represented here, and MODIS, which are the pixels represented here. This is a MODIS time series, so you can see there's tremendous fluctuation from year to year in the timing of the production as well as the actual magnitude of the greenness value, the highest greenness value. So we're bridging these data sets, and we have more work to do in this arena, but this is where the data sets are going to come together. So next steps, one of the two things I wanted to highlight is the fact that we can replicate the analysis that we conducted with the phenocams because they're using a standardized protocol. They're in the phenocam network. So the same analyses that I showed can be done at any of these sites that are represented across the United States who represent agro-ecosystems. I'll highlight a quick example from Walnut Gulch. So the graphs look a little bit different, but it's the same data that we were looking at with the green lines in the earlier graphs. So these are greenness responses from layman's love grass in the top panel and creosote shrubs in the bottom panel. There is an eddy covariance tower co-located with these observations, so we can link it to carbon flux, as well as to kind of enhance our interpretations of the factors that are driving these patterns. Secondly, we're engaging citizen scientists. So this is a really exciting effort through the National Phenology Network. We're working in partnership with them, and the season is the campaign is called Southwest Season Tracker. And the idea here is to, one, enhance the understanding of ecology and specifically phenology to the general population. And secondly is to motivate them to collect data that we can then use to verify our model outcomes. So if we go back to the very beginning, we talked about the mechanistic understanding of the factors driving plant phenology in these regions, so that we can forecast or predict when things will happen and how changes in temperature and rainfall can influence those patterns. So this is an exciting campaign that just came online about a month ago, so if you're interested, you can join us here. But we'll be leveraging these data to verify models that are yet to come. So a lot of people involved in collecting these data and this effort, and I want to just briefly acknowledge them here, and I really want to thank you for your time.